Today we will be discussing the host immune response of Epstein-Barr virus. As previously discussed in my last video, Epstein-Barr virus is transmitted through the saliva and finds its way to the oropharynx epithelium. Here the virus crosses the epithelial membrane and finds its main target, the B lymphocyte. The B lymphocyte expresses a receptor called CD21 and co-receptor, which consists of MHC2 molecules. The CD21 complement receptor of the B cell will bind to the viral glycoprotein 350 on the EBV viral particle. It is still unknown whether the viral replication initially occurs prior to entry into the B cell or once inside. But once inside, the EBV begins replication, and the virus produces over 80 new proteins that code for new viral particles or immune evasion. The mechanism by which lytic proteins evade the immune system is by interference. Either they interfere with antigen presentation or processing on CD8 T cells, or they may also downregulate the MHC class II expression. Immune system evasion allows for Epstein-Barr virus to establish infection within the individual. EBV is classified as a persistent infection because of its latency characteristics. Epstein-Barr virus can establish latency in a number of ways, one of which being able to produce a variety of latent proteins within the B cell, along with small coding and non-coding RNAs and microRNAs. This is known as latency 3, as seen on the far right, and commonly leads to proliferation of infected cells. In the middle, we have latency 2. EBV, nuclear antigen 1, and latent membrane proteins are expressed. On the second to left-hand column, we have latency 1, which is when only EBV nuclear antigen 1 is expressed. And then finally, we have latency 0, where expression of all viral antigens are silent and usually unable to be detected. Latency 0 is commonly found within the blood of healthy individuals and can circulate between the lymphoid tissues and circulatory system. Now we will discuss the innate immunity of lymphocytes in the Epstein-Barr infection. Primary infection of EBV has been concluded based off studying patients who have had infectious mononucleosis. Innate immunity is carried out via natural killer cells. Natural killer cells have various responses during EBV infection. Infected B cells activate NK cells via EBV-induced ligands. Once the NK cells are activated, they can decrease EBV-mediated growth, transformation, and proliferation of B cells. Failure of natural killer viral control could lead to infectious mononucleosis. Moving on to adaptive immunity, during primary infection, the number of CD8 T cells increase 5 to 10 times higher than normal. Once the infected B cell travels through the circulatory system and into the lymph nodes, this stimulates CD8 T cell activity and binding. CD8 T cells go on to differentiate into cytotoxic T cells, which will then circulate throughout the blood, tissue, and lymph. EBV also activates CD4 T cells, which will then differentiate into CD4 T helper 1 cells and produce specific cytokines for EBV and eventually activate the B cells. The activated B cells will transform into plasma cells and release the specific antibodies against EBV. B cells participate in isotype switching, meaning that they first create antibodies specific for EBV viral capsid antigen with IgM, then with the help of T cells, switch to produce antibodies for EBV viral capsid antigen with IgG. Finally, the plasma cells will produce Epstein-Barr virus nuclear antigen, IgG.